Hello, this is Joseph Kaziri, instructor and lab coordinator here at Mississippi College in the biology department. And today, or in this video, we are going to discuss C-fern genetics. Now, C-fern genetics, we're going to separate this into two parts. The first part, I'm going to talk about the genetics, and then we'll come back and discuss the C-fern and its life cycle. Because the C-fern in this life cycle is something you're going to be noticing, and we're going to come back to in Biology 112. All right. Before I forget, on page 26, you need to be familiar with those terms and what they mean, because you will see them again. Anything we pick up in this class, you're going to be covering portions of this in your lecture. Okie doke. So for starters, let's talk about genetics. All right, it started off with Mendel. Mendel in the mid to late, I think 1860s, 1890s, I can't remember. Gregor Mendel was a monk, but he was also a mathematician and a botanist. So he did a lot of studies with his pea plants. You know, being a botanist, having that kind of resources, he studied several different traits. Right, the trait of height, flower color, pea color, pea texture, and each one of these traits, they have at least two phenotypes. Phenotypes is how we describe organisms. When he did this, he did it with a little less than 2,000 plants. He looked at the flower color. Was the flower purple or was it white? He looked at the height of the plants. Is it short or is it tall? He looked at the peas. Are they yellow or are they green? Are they wrinkled or are they smooth? Right. All of these observations he could easily make. So let's refer to a few of those. First, let's discuss a few times we're going to see P, F1, and F2. This stands for the parental generation, sometimes pure. All right, the first generation offspring, second generation of offspring. And the phenotypes that we're going to discuss here are for height, tall, and short. Before Mendel, they didn't really know about genes, but he got an idea of it. Well, just like any math problem, when you want to start any problem, you need to start with something you know. So what he did here is he didn't just take a tall and a short plant. He crossed a tall plant over and over again until it produced all tall offspring. Same thing with the short. He took short plants, crossed it with other short plants, the pea plant that is, short pea plants, until it produced nothing but short plants. So it only had tall and short genes. So this is a purebred tall and a purebred short. He crossed those together. So the pollen, it's the male parts of the flower with the female parts, produced a seed, and he noticed that all of the offspring, all of them, 100%, were tall. Now, looking at that issue, we see that, well, maybe the tall just takes over and the short disappeared. Right? But he went on. He crossed one of these offspring from this generation with another. So he inbred them, crossbred them, however you want to refer to them. And he noticed that, he observed, you had tall and you had short plants for this pea plant. So obviously the short didn't disappear. The short has been there all along. But being a mathematician, it wasn't just tall. Most of them are tall. In terms of a little less than 2,000, I think it was like 17 or 1800. Right? they noticed that there was a ratio of 3 tall to every short 1. And once again, right, using his powers of deduction, being a mathematician, he came to the conclusion you could only have this type of ratio if there were 2 characters inside this tall plant and 2 characters that determine the short plants. So let's use 2 characters. We assumed, and first of all, let's go ahead and define some letters. We're going to assume T, a capital T, is the character for the tall plants, and a lowercase t is the character for the short plants. So this tall had 2 capital characters, which mean for tall, because it was purebred, and the short had 2 shorts. We're getting into something called the genotype here. The genotype is what you can't see. It's the arrangement of genes. And yes, you do have 2 alleles for your height. One comes from your mom, one comes from your dad. This is usually true in sexually reproducing organisms. Let's go through the same scenario, but with the genotypes. When people ask genotypes, they're looking for letters. When these 2 are crossed together, you got one large, one small. This is a heterozygous genotype, where this would be a homozygous and another homozygous. Homozygous dominant, we'll refer to it from here on out, and homozygous recessive. Anytime you cross homozygous dominant and homozygous recessive together, you're going to end up with all, this is going to be 100%, heterozygous genotypes. Well, if you had these 2 characters and they gave you a tall organism, that tall character must somehow mask the other. So the capital letter is representative of the dominant character, the dominant allele. But we have a lowercase, which is a recessive allele, which can be hidden. Sometimes a recessive allele, that could be referred to as a carrier. All right. So what he did is he crossed a heterozygous individual with another heterozygous individual. You come to find out, well, it doesn't stop there. You would end up with one homozygous dominant, 2 heterozygous, to 1 homozygous recessive. Now, if this 1 and that 2 right there have the dominant alleles, means that would be your tall. That's your 3 tall to your 1 short. Now, how did we get this first number? This first genotype and the second genotype? Well, we use a phenotype for that. A Punnett square to determine the genotype. So what he determined is that these 2 characters, we don't know which one of these are going to fertilize the next offspring. Somehow they must separate. And that leads us to, as they're separating, to the law of segregation. Which means we have these 2 characters inside an organism. We have these 2 characters over here. Doesn't really matter where I put each one. But during its life cycle, when it's trying to produce some little gametes, when it's trying to produce offspring, these are going to separate. Where we don't know if let's say if we're talking about sperm, is the sperm going to have this dominant allele? Or is it going to have this dominant allele? In the other organism, since we have these 2 recessives, they're also going to separate. All right, let's say if this is in the egg cell. We don't know if the egg cell is going to have that allele or it's going to have that allele. So what we're doing here is we're describing a little process called meiosis. Looks like meiosis. All right? Mendel was describing meiosis decades before it was later described by saying the law of segregation. Right, that means meiosis is occurring. It means your DNA has to separate to make your sex cells, your gametes. And every time you get a Punnett square and you put those letters, you separate them over or on the side of those of that grid, you're confirming, yes, meiosis does occur. All right, you're agreeing with the law of segregation. Now, this is why this only works for reproductive organisms, because asexual organisms, right, they don't separate. They don't make gametes like we do. No eggs and sperm. So we don't know if that dominant and that recessive allele will come together being fertilized. We don't know if that one and that one will come together. That one or that one. Big T or that little t. Right. Now, inside is the possible fertilization possibilities. Possible fertilization. Every one of them are heterozygous in genotype, which we see here. That's the only thing that can occur, because anytime you have two homozygous, a dominant and recessive together, cross together, you will get 100% heterozygous. What would that look like? Dominant, 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 dominant. That'll look all tall. All right. Well, when those guys crossed again, let's do another table. He crossed a heterozygous genotype with another heterozygous genotype. All right, we just separate them, one allele, one gene, one allele over each grid. We don't know which. Now, we just agreed that meiosis, the law of segregation, occurs. But we don't know what egg and sperm will come together to fertilize. It happens in such a large number, it's kind of beneficial to use plants. We don't know if this dominant allele from up here will fertilize with this dominant allele. We don't know if that dominant allele and that recessive allele will come together. I'm just putting the dominant first, because usually that's what looks best. This dominant and that recessive, or that recessive and that recessive. Now, if we put this in terms of a ratio, we see we get one homozygous dominant for every two heterozygous genotypes to every one homozygous recessive. Right. This right here is referred to as your, genotypic ratio. Or this down here refers to the phenotypic ratio. You know, at this point, every time you have two, when you cross 1, 2 heterozygous together, you always get a 3:1 phenotypic ratio, or a 1 to 2 to 1 ratio. When you do ratios, you have to say tall or short. You cannot just say 3 to 1, all right, because you're not telling me what you're dealing with. So these are the possible gametes. Now I'm going to continue with page 28, which discusses the dihybrid cross. Because what we just seen here is a monohybrid cross. That is a cross when we discuss just one trait. What trait? The trait of height. Right? That's height and height. We're not looking at height and color of the flower. No, if we're looking at flower color, that's one trait. Monohybrid cross is when we look at individuals who differ only by one trait. So it's basically two forms of that trait. Okay. Now let's talk about, going from a monohybrid cross, let's talk about a dihybrid cross. All right, when you talk about more genes, you got to include more blocks. All right. Now, dihybrid cross is when we're talking about two traits. All right. And let's use the traits. Let's see. We're going to use height and P color. So we got a big T and we got a little t, which is going to be for the tall, just as we've seen before. Little t is the character, the allele for the short plant. So that's your height. Right, and then P color. Now, is the P going to be green? Or is it going to be yellow? Right. Where green is the dominant allele. So if you have a big G and a little g together, it'll be shown as green. All right, so there's your little legend. Right. So what Mendel did is he did two things. All right, let's do this just as we did before. For parental generation, first generation offspring, next generation. He took a tall, let's see how to set this. He took a tall plant that produced all green peas, right? And he crossed those together again and again and again till he only got to that plant only produced tall green offspring. So we can give that a genotype, but let's come back to that. And then he did the same thing. He had a short plant that only produced yellow peas. Whoop, kind of mixed up what I was doing. You'll see what I'm getting at with that. Short with yellow peas. Bred them together, they're inbred. They're now the purebred ones. So according to what happened up here, right, whichever one is dominant, when you have two of them that are purebreds, two homozygous, or better right here, you will always get heterozygous. So we got this is double homozygous. I guess I'll go ahead and put it down now. Put it right here. Double homozygous, big T, big T, big G, big G, and he crossed it with short, which is a double homozygous recessive, little t, little t, little g, little g. Right. And if we cross these together, then we're going to get a double heterozygous. What would that look like? Well, T is for tall, big G is for green, so we're going to get tall plants that produce green peas. How many? 100% of them. Where's the short yellow? They'll come back. All right. So we're talking about the phenotype over here. Here's our genotype. Right. Double homozygous dominant, double homozygous recessive, double heterozygous genotype. Now we have two genes without G for T. All right. What he did next was he crossed two from this generation together. All right. Now this is where it may get a little confusing because we have to put the possible possible alleles up here as well as on the side. I'm going to do it this way. Let's use a green for this. Big T, little t, big G, little g. All right, when these separate, Mendel was curious to find out. We know that these separate up here, but what if you have two genes? All right, the tall, the height, and the color gene. will they always stay together? Will the tall green plant will tall always produce green plants? Will short always produce yellow plants? Because as of right now, it looks like it. The tall always produce green. But we have to get to another generation to figure that out. That's like assuming just because you have blonde hair, blue eyes, that's the only option. Blonde hair only goes with blue eyes. But that's not true. You can have blonde hair and have brown eyes. We don't know which of these will come together. Mendel was trying to decide. He says, if that was true, then I'm either going to have tall green. One of his hypotheses here. Hypotheses. So you have tall green and short yellow, or he's going to have a spectrum, right, of everything. So let's find out which one is correct. So in doing this, we don't know if this dominant, this big T, and that little g, switching colors on you. This big T and the big G will go into an egg or sperm cell. Right? Maybe that big T and that G might combine and be in an egg or sperm cell together. Or the lowercase t, right, can separate and join with, maybe stuck with that big G. I'm just putting the T's before the G's. Or will those two come together? This is how you're going to figure out your possible gametes for this dihybrid cross. And you should be familiar with this, or at least seen it once or twice, because you're going to be using it in a couple of labs. Now, since this parent and this parent are the exact same, we can assume that this goes the same way over here. Big G, big T, big T, little g, little t, big G, little t, little g. Right. Now we need to combine them. Right. Remember, those are your possible gametes from the parent. This is your possible gametes from the other parent. These are all the possibilities that can occur in fertilization. We're trying to figure out, are we going to get all tall green, well, either tall green, short yellows, or are we going to get a spectrum of everything? Well, this is the time when you fill it out. So I'm going to put the big T, big T, big G, big G. I'm going to move through, then I'm going to fast forward this. All right, these two come together. You got big T, big G, little g. All right. Let's see, big T, little t, big G, right, big G, big T, little t, big G, little g. I'm just bringing them together. All right, and what you should be doing is you should be doing this too. I'm just going to pause it, and all of a sudden it's going to come right back, and it's going to be filled. All right, now we're back. All right, I filled mine up, put all the possibilities out there. We're not going to come up with a, what should I say? Genotypic ratio. We're going to come up with a phenotypic ratio, because the genotypic ratio would be just a little more difficult. So we're trying to simplify it. So let's talk about what each one of these are, and I'm going to pause it again. Big T, big T. This shows that you understand what's going on. If you can look at this, you know the legend over here. Looking at this, and you know that's a tall green plant, or excuse me, tall plant with green peas. All right, for example, even over here, it's a tall plant, right, with green peas. Start moving down here, you got tall plants, right, with yellow peas. Here's a short plant with green peas. Here is a short plant right, with yellow peas. Right, so you're trying to figure all this out. Right, you determine the phenotype from the genotype. So what you need to do is you also need to be working on this. You need to finish that up. And I'm going to fast forward this, and I'll come right back with it done. All right, now that we determined the genotype for each one of these, let's figure out what our phenotypic ratio would be. Looking at all of this, we have, let's see, well, what are the combinations? The spectrum, which I was referring to earlier, is going to be, let's see, you might get some tall greens. All right, tall greens. We might have some tall yellows. We may have some short greens. And we might have some short yellows. In terms of the tall greens, if you count them up, 1, 2, 3, 4, 5, 6, 7, and missed some. 8, 9. There we go. 9 is for tall yellows. We have tall yellow 1, tall yellow, tall yellow. You have 3 tall yellows, short and green, short green, short green, short green. That's 3. Short yellows, there's only 1. All right, so here's our ratio. You got 9 tall greens to 3 tall yellows to 3 short greens to 1 short yellows. If you add that up, that equals 16, which means there are 16 squares up here. So what you need to know, right, when you cross 2 heterozygous genotypes together, you always get a 3-to-1 phenotypic ratio. Here, when you cross 2 double heterozygous genotypes, you will always get a 9-to-3 to 3-to-1 phenotypic ratio. So between these two hypotheses that Mendel proposed, right, either all the offspring are going to be short, yellow, or tall and green, or you're going to have this variety of all of them. Well, obviously this was not correct, right, because what he did notice, he got of that same ratio. All right, we're going to stop here, but then I'm going to pick back up with a second video on the C-ferns.